Welcome to Chapter 12. So this is a great follow-up to Chapter 11 because as you think about ways of decontamination, our antimicrobials or our antibiotics do the same process of cleaners. So they're going to target the same microbial components. And so we actually hijacked our antibiotics from other fungi and bacteria. In particular, streptomyces and bacillus bacteria are huge contributors, so streptomycin, if that sounds familiar to anyone. And of course, penicillium, so molds such as Penicillium and Cephalosporium, they inhibit the growth of bacteria because they're competing for space and nutrients. And so when they're in the same habitat, this is a way for them to adapt and survive. And as humans, we have used those to our benefit and possibly even to our demise. And so penicillium is how we have our various penicillins. Bacillus species produce things like bacitracin and polymyxin B. And of course, streptomyces species, we've gotten quite a few various antibiotics from those. So the interactions between the drug and the microbe have to be this perfect balance. They need to be selectively toxic. And so a way for me to illustrate this for you is when you think of a person who is unfortunately having to undergo chemotherapy. So the radiation or chemotherapy is not a selectively toxic process. It primarily is targeting the most rapidly growing cells such as cancer cells or bacterial cells if you have some sort of infection. But as a consequence, you also have a loss of your own cells, which is why people undergoing chemotherapies and things like that are often very sick. They're very immunocompromised. And so when we think about how to design these medications to be selectively toxic, it's really quite a challenge, in the sense that you have to target things that are distinct and specific to the bacteria or the microbe that the human body does not have. And so a good example of something that is not superbly selectively toxic is that of the ribosomes. So if you have a drug that distinctly inhibits bacterial ribosomes, although the bacterial ribosome is different than ours, so you can see here on the right, you've got the 70S unit versus the 80S, which is what we have. But ultimately, the size and shape are about the same, and so as a consequence, you end up having side effects, so nausea, vomiting, discomforts, malaise, those sorts of things take place because of the fact that the drug may not be as selectively toxic as one would prefer. And so various mechanisms of drug action, this is a great overview for some of the specific drugs that we'll talk about through this chapter. And in particular, cell wall inhibitors versus cell membrane. So cell wall inhibitors, things like penicillins and cephalosporins are going to fall in that category. Cell membrane inhibitors, so things that are going to interact with the phospholipid bilayer. DNA and RNA, so actually inhibiting replication versus protein synthesis. And so there's various drugs that target different parts of the distinct ribosome for various microbes. And then of course metabolic pathways. So as a bacteria is undergoing normal metabolism, it is important to be able to target that bacterial metabolism and leave host cells unattended. And so the example that we'll talk about that is with sulfa drugs. And so our drugs can be either narrow or broad spectrum, and so narrow spectrum means that it is a drug that is only going to target one class or type of bacteria, so a gram-positive bacteria versus gram-negative. A good example of this would be a drug that targets only lipopolysaccharide, because lipopolysaccharide is only found in the gram-negative cell wall. Broad spectrum, on the other hand, is going to target things like ribosomes. And so ribosomes, as I showed you before, are very similar among eukaryotes and prokaryotes. And so as a consequence, you have a wider range of reactivity, and unfortunately you can in turn have a wider range of toxicity. So our penicillins and cephalosporins are going to target the bacterial peptidoglycan, and so these are going to be most effective against gram-positive bacteria because of the outer wall of thick peptidoglycan. These drugs have been modified over the years because they are one of the most predominant types. And as a consequence, we have moved them from being a narrow spectrum to becoming broad spectrum, meaning that they work well against both gram-positive and gram-negative bacteria. And so the activity of peptidoglycan is illustrated in the MRSA case study, which you'll have that activity available to you. And so what you find is the specific bacteria, as they're laying down the units of peptidoglycan, there's an enzyme that creates the peptide bonds that form between each of these repeating units. And so that enzyme is the PBP, or penicillin binding protein. And so it's named PBP simply because that's how they discovered it. But ultimately, what the enzyme's purpose is a peptidoglycan binding protein, really. And so the PBP protein binds these peptidoglycan units and creates peptide bonds. And so that is bonding. So think of this as like a brick wall. So if these are each little bricks, and these little amino acids going between the bricks are like the mortar. And so that keeps that brick wall intact. And so if you inhibit that process, what happens then is the bacterial cell walls will lyse. And that is exactly the mechanism of action of a beta-lactam antibiotic. A beta-lactam antibiotic is nearly half of all antimicrobial drugs. Penicillins and cephalosporins are beta-lactams, and they're really one of the most predominant and most proliferatively used. And so what exactly is a beta-lactam? Well, it's actually a chemical structure called a beta-lactam ring that they all share. And so this is the backbone. We call this the backbone of the chemical structure. So all of the beta-lactam antibiotics have both the beta-lactam ring, the thiazoline ring, and of course the variable side chains. The side chains are found here. And what those side chains do, they allow for different reactivity. So a side chain can make the difference between a 4-hour acting medication and a 12-hour acting medication. Or it could be the difference between a medication that requires 4 doses a day versus, only a couple. And so you can impact longevity, you can impact the frequency of dosing to reach an effective dose based on the interaction of those side chains with your cells. So cephalosporins fall in that same category. So you'll see that imidazole ring shown here. And then you have 2 R groups, so 2 functional groups on both sides. And so cephalosporins are essentially the synthetically altered version of the beta-lactams. And it's been modified pretty successfully in the sense that they have 4 generations. So the first generation cephalosporins are pretty narrow range in the sense that they target distinctly gram-positive bacteria. And after successful generations, they were able to make this a broad-spectrum antibiotic. And so what this did for the clinicians is it allows them to treat polymicrobial infections with a cephalosporin, so if it has both gram-positive and gram-negative. And then again, it also allows them to treat infections with a broad-spectrum cephalosporin when they don't actually know what type of infection is taking place. So this is both good and bad. So it allows for broader coverage and protection, so the doctor can have less information and still treat their patient. But inevitably, if you're using a broad-spectrum antibiotic and you only had a gram-positive infection, you're then wiping out normal flora. And hopefully by this point in the semester, you've realized that normal flora is part of your immune system. It is part of the first line of immune defense. It works very closely with your body, maintaining homeostasis, so your pH in your vagina, by maintaining coverage, so it inhibits the growth of other microbes that would cause you disease or harm. And so when you treat with broad-spectrum antibiotics, you're wiping out not just the infective agent, but you're also going to wipe out your normal flora, which is why, of course, women tend to get yeast infections upon taking various antibiotics. And so non-beta-lactam cell wall inhibitors, the example of this is vancomycin, and the MRSA case study illustrates this very well as well. And so in the MRSA case study, you can see that vancomycin is binding distinctly to the amino acid chain on the peptidoglycan wall. So where the beta-lactam antibiotics bound to the enzyme, vancomycin is binding directly to the amino acid chains. And so the mechanism of action is the same. It's still inhibiting cell wall, but the way that it inhibits the cell wall is slightly different. And so vancomycin is very narrow spectrum because it's only going to bind to certain amino acids. So staphylococcal infections that have become resistant to penicillin and methicillin are now treated with vancomycin. Vancomycin, it's interesting. The book provides the fact that it's toxic and hard to administer, but usage in the clinic now, if any of you are already in the clinic, you'll realize that vancomycin is actually used quite readily and quite broadly. And part of that is because of the reduced access and efficacy of antibiotics because of the way we've been using them in the years past. So the other non-beta-lactam cell wall inhibitor is bacitracin. This is taken from Bacillus subtilis. That is a bacteria that does make endospores, by the way. And so essentially what was taken is this is used as a topical ointment. So Bacillus subtilis produces a substance that allows then the interaction with the cell wall and the breakdown. So it allows Bacillus subtilis to enter the body. And so instead of allowing Bacillus subtilis to take that, we monopolized on that capability and created the antibiotic bacitracin, which we use as a topical ointment. So we're now moving to the cellular membrane. And so the cell membrane, in the previous chapter with chapter 11, think back to the surfactants and how they were kind of pushing apart the phospholipid bilayer. The same thing is happening here. So polymyxins are going to do that same sort of process. They're going to push those phospholipids apart, and they're going to cause cell membrane leakage. And so same sort of idea with amphotericin B. They're going to push those phospholipid heads apart, and so the fungal membranes are then going to have leakage. And that's shown here. So polymyxins interacting with the surface layer of the phospholipid heads. Those interactions then push the phospholipid, and then membrane integrity is broken down. And of course, the osmotic pressures cause the cell to burst. Drugs that impact protein synthesis, just as a reminder, all drugs that interact with protein synthesis are going to be broad spectrum. They're going to act pretty effectively against a wide range of microbes that have proteins, and that's pretty much everybody. And so aminoglycosides, such as streptomycin and gentamicin, when they're acting, they actually cause a misreading of mRNA. So when protein synthesis is going through, it causes it to be blocked because it's not reading the strand in the correct order. Tetracyclines, they're actually going to block T for tRNA, so tetracycline T for T, and so they're going to block the transfer RNA on the A acceptor site. So they're going to pretty much completely shut down protein synthesis. Those tetracyclines are going to be broad spectrum. So treatments for things like various STIs, Rocky Mountain spotted fever, Lyme disease, so huge, wide range. And let me draw your attention to the fact that they're also going to treat protozoa, which are eukaryotic parasites, such as Trypanosoma cruzi, Trypanosoma brucei. Those can be treated with tetracyclines because the ribosome is so similar between prokaryotes and eukaryotes. Cost is very low. It does have some toxic side effects. So tetracyclines, have side effects in the patient. And so broad spectrum, meaning it's going to act not only on gram-positive and gram-negative, but actually on prokaryotes and eukaryotes. Drugs that are going to impact metabolism can act a lot like various inhibitors. So by interacting with metabolism, you are actually stopping the microbe from obtaining the nutrients and energy needed to survive. And so this can take place by inhibiting directly the enzyme active site of an enzyme necessary for metabolism, so that's competitive inhibition. Or you can use multiple drugs and do a synergistic effect. So often we have combination drug therapies for very difficult infections. And so then they're going to interact with both various parts of the metabolism, but it may hit in different sections of that process. And so sulfonamides are the example of a drug that interacts with the metabolic pathway. And in particular, it's going to block the ability of the bacteria to use folic acid and make folic acid. And so the image down at the bottom, so you can see that the normal substrate, the PABA substrate here, binds to the spectral enzyme. But what the sulfa drug does, it is actually a competitive inhibitor. And so instead of allowing the PABA to bind, it actually then allows it binds instead and takes the place of the PABA. And so sulfonamides inhibit that enzyme, and then trimethoprim can inhibit the enzyme later down the road. So these two used together are synergistic. So what you would find is that you're going to have much less bacterial growth in the combination therapy of sulfonamide and trimethoprim than you would with one drug only. So those are various examples, things that they could potentially treat. So drugs that impact nucleic acid synthesis, they can end up blocking the nucleotides by actually acting like a nucleotide analog, or they could block the enzymes that facilitate replication. And these are going to cross the gamut. So these are going to be impacting both prokaryotes and eukaryotes. An example of a drug that blocks nucleic acid synthesis is chloroquine. So chloroquine is commonly used to treat malaria. Malaria is a protozoa, so that is a eukaryotic microbe. It is an infection that is endemic to sub-Saharan Africa, where a distinct mosquito exists. And so this drug causes a cross-link. If you think back to genetic barriers, you'll see back in chapter, I think it was chapter 14, when we were talking about genetic barriers, so the spread of malaria, so malaria impacts our red blood cells. And so patients or people in that area typically have sickle cell anemia, which prevents malaria from entering. And so there's both genetic changes that have taken place to survive malaria, as well as we have chloroquine for people who can afford the drug going to visit those sorts of areas. Quinolones inhibit DNA helicases, and just as a quick refresher, the DNA helicase is shown there. It's actually unwinding the double-stranded DNA molecule. And so DNA helicases, examples include Cipro, levofloxacin, and so the quinolones are shown down there at the bottom, and they treat a huge range of infections. So they have a broad range of usefulness. In particular, I think of Cipro because it was the first line of treatment against anthrax when we had the anthrax scare about a decade ago. Antiviral chemotherapeutic agents. And so these antiviral chemotherapeutic agents are really amazing. We insert parts of acyclovir, or ribavirin, or AZT, and so it looks like we're providing normal thiamine or normal guanine, and it actually impacts and blocks the replication of that virus. So the other ways that these drugs can act, it can block penetration, and so penetration is, those first steps. So you have absorption and then penetration into the cell, and so steps 1 and 2 of viral replication. And by blocking that process, if it can't get in, it can't infect. And then, of course, maturation of the particles. Maturation takes place when you're moving through the assembly step, and then, of course, the budding off or exit or lysis on that cell wall. And so being able to block the maturation, you've sacrificed that one cell, but ultimately you've saved a whole host of cells within the patient's body by being able to prevent the spread of those viral particles. And then protease inhibitors, a good example of that is from the video with the neuraminidase, which is so much fun. So neuraminidase is a protease enzyme. It cleaves various proteins, is essentially the general term for a protease. And so in particular, neuraminidase cleaves the attachment proteins on the cell surface. And so being able to inhibit this protease in viruses such as HIV and flu, you can actually inhibit their ability to attach and exit to the antiviral chemotherapeutic agents. So at this stage of the game, you're dealing with viruses and you're dealing with our DNA and RNA sequences. And viruses have been responsible for lots of changes in human evolution simply by inputting small segments of viral DNA in our bodies. And so when you come to thinking about toxicity, it's really quite difficult to create a selectively toxic drug when you're dealing with a chemotherapeutic agent. And so the various drug actions that we're going to talk about, it could block penetration of the virus to the cell, so if it can't attach, it can't infect. They could block replication, so the drugs that we use are often either guanine or thymine analogs, and so we prevent the usage of our host cell machinery by those viruses by putting in like a decoy. We can block maturation. So maturation is when you're going through assembly and release in the viral replication process. And so by blocking them being able to get out of the cell, you're blocking the spread of the virus from an infected cell to neighboring host cells. And then protease inhibitors, neuraminidase is an excellent example of a protease. And so hopefully you enjoyed the flu video. And so a protease inhibitor is going to block the ability of this protease enzyme from cleaving other proteins. And so in this case, neuraminidase is needed to be able to cleave its way off of the host cell membrane. And so by blocking that cleavage action, you're keeping the flu virus on that one cell and not allowing it to spread to other cells. So drugs for treating flu, amantadine or rimantadine are pretty exclusive to both influenza A infections. They're going to impact the fusion of the specific virus with the cell membrane. And so you're looking at that process up here, so being able for absorption. And then Relenza and Tamiflu, these guys are slightly broader spectrum, and they're going to block neuraminidase. And so they're going to block the ability of the neuraminidase protein to cleave, because neuraminidase actually does cleave here upon absorption, and then it cleaves here upon exit. So it allows it to be separated from the cell membrane because it binds and attaches, so it's cleaved off here, and then it binds and attaches, and it has to be cleaved off to be released. Anti-herpes drugs such as acyclovir, there's a whole host of these drugs. Acyclovir is the predominant one that you should consider knowing. And what they're doing is they're terminating the DNA replication. So they're blocking the replication process. And so they're competing for replicating DNA in the virus, so it turns the process off with an analog. So HIV and AIDS, the big HIV drug that you want to consider is AZT, so azithromycin. It is a thymidine analog, and it's been very successful. It interferes with the DNA sequence because of using a transcriptase inhibitor. Interferons are secreted by our body as a second line of immune defense, and these interferons are really important for activating immune cells so that you have a robust immune response and you don't allow viral or cancer escape. And so what we have done, therapeutically speaking, is they've isolated interferon. And so when you have patients suffering from certain conditions such as chronic hepatitis or leukemias or cancers, you can actually give them interferon to help their body treat those sorts of infections. And so here's an example of interferon beta, and so potential side effects and things like that, and of course all the contraindications. But ultimately being able to take someone who's suffering from genital warts and put them at least in remission for a period of time is quite worth some of these side effects. So it's kind of that balance that is struck. So newly developed classes of antimicrobials, ketolides fall into this category. They're very similar. They're kind of a modification of erythromycin. They're going to be used against gram-positive infections, especially when those are resistant to existing macrolides. And then linezolid, I call it linezolid because I have a colleague, we published a paper on this. Oxys, that's just a mouthful, so nasal it is. And what you find is this antimicrobial is going to block mRNA, so it's going to be broad spectrum because it's interacting with the ribosome. And it's used to treat respiratory infections. The study that we ended up doing, we found that it was actually very effective at preventing secondary bacterial infections after influenza infection. It can treat MRSA, VRE, so vancomycin-resistant enterococcus. And so it's a very a useful drug, and we're learning a lot more about it. So the acquisition of drug resistance, just as a reminder, I know we've talked about this before, but just to bring it kind of back home, remember that first and foremost, the microbe changes. It's not the drug that you give the microbe. It's the fact that the microbe is constantly evolving, just like we evolve, just like animals evolve. And then what happens is we put selective pressures. So if you have one microbe that has obtained resistance and it's in a colony is susceptible, once you give the drug, you wipe out everyone that's susceptible, all these bacteria that can't survive that antibiotic, and that then gives space for the resistant bacteria to thrive. And so this is more of a vicious cycle that we're creating. And so the exposure pressures, the loss of those, you end up having that loss of normal flora. The drugs leave you immunocompromised. You may have other comorbidities such as obesity, diabetes, heart conditions that make us all much more susceptible to these sorts of deadly situations with infections. And so ways that bacteria can transfer DNA, plasmid, we talked about conjugation. We did not talk about transformation, but it's where bacteria can pick up free pieces of DNA. And then viruses, such as bacteriophages, can insert bits of DNA. And so as a consequence, the ability of these microbes to share genetic information back and forth allows them to directly evolve and change. And it's actually the human decisions that are causing these antibiotic or chemotherapeutically ineffective strains of viruses or bacteria. And so here is that example. So it's showing you that you've got a whole colony of cells. You happen to have a mutation in one cell, and then you expose those to drugs. We go to the clinic and we say, hey, I need antibiotics, and you actually have allergies. So you see that everyone that's susceptible dies, and then you have this outgrowth of the resistant. And so it's natural selection at its finest. And it's brought about and it's propagated by poor decisions. And so as a consequence of those poor decisions, we have superinfections. We essentially have really, really nasty side effects to some of these drugs. You can have allergic reactions. There's direct toxicity. So if it's a broad spectrum drug, toxicity, in the liver and the kidneys. And then of course, when you disrupt your normal flora, you have superinfections. So it's not just what you're trying to treat, but then now you're going to have multiple other infections emerge from opportunistic microbes that already exist on your body. And so the great example of a superinfection is, of course, Clostridium difficile. You can see that the intestine normally perfectly fine, but then you potentially wipe it out with a medication that maybe is treating an upper respiratory infection, and then you have a superinfection in your gut. So this pathogen is just going to completely overgrow. And in the case of Clostridium difficile, it's a really big challenge because it's resistant to most antibiotics. And so you're looking at either therapeutically treating it with a cocktail or moving to new age medicine with fecal transplants. So which of the following effects do antiviral drugs not have? Killing extracellular viruses, stopping virus synthesis, inhibiting virus maturation, blocking virus receptors. Viruses, by their definition, are obligate intracellular parasites. So the best answer is A. There are no extracellular viruses. A drug that targets lipopolysaccharides and the gram-negative cell wall would be considered broad spectrum, narrow spectrum, selectively toxic, broad spectrum and selectively toxic, narrow spectrum and selectively toxic. So it's only targeting lipopolysaccharides. So the best answer would be narrow spectrum and selectively toxic. So aminoglycosides are antimicrobial drugs that target which of the following? Peptidoglycan synthesis, cell membrane, protein synthesis, or DNA replication. Best answer, protein synthesis. Which of the following drugs inhibits bacteria by blocking folic acid synthesis? A. Chloramphenicol, erythromycin, sulfonamides, tetracycline. Sulfonamides. So I know this chapter has a lot of information. Try your best to distill it down. This may be a chapter that note cards would be very helpful. And kind of put it back into the global context of where does this drug act? Why does it matter? So is it acting on protein synthesis? Is it acting on the cell membrane? Is it acting on the cell wall? If it is a viral antiviral drug, where exactly in the replication cycle is this drug acting? Is it inhibiting, the synthesis step, so step 4 of the replication cycle? Is it inhibiting virus entry? Is it inhibiting virus exit? And so those diagrams should kind of help you with that process, but try to connect those dots and connect them back to especially chapter 6 with viruses. And then where does it act in the same regard like with physical and chemical agents? Reach out if you have any questions. I'm always here to help.